Hello, good afternoon. So let's continue on then with the class. Yesterday or on Tuesday we went on to, which I was starting with the wrong class, then I corrected my course pretty quickly, and then we started with fertilization. So we continue on then with the fertilization side. So we talked about a little bit about all that process of capacitation and acrosomal reaction. Okay, all of those things, if you take, and up to this point for this class, we are roughly midway through the classes. It's not difficult, you know. It's really not difficult, all this stuff. The only thing is a lot of little things. So eventually, you're gonna get out of this class at the end of December, and you're gonna know a lot of new names, a lot of new little processes, a lot of these things. So. But as long as you have this bigger picture, and that's why, you know, you asked me, you know, last time, you know, are you going to this slow and fast, you know, block? But no, we're not, because otherwise we start getting too much detail of these little things. Important, but on the overall picture, smaller things. So on the oocyte side, we went on. I think we stopped at block of polyspermia, which we actually already discussed before. But basically speaking, like, and this is probably the one of the classes that has the most little tables in the slides. Okay, I included lots of tables because they are indeed very nice. If you go and read column and what it is in the next, well, what is the process, and explain a little bit. It is actually really nice little table for you to read. Okay, it gives you quite a bit of information. It matches really well what we saw in class, so it's a good material for you to study. Okay, we mentioned this thing changes. What is the importance of having, you know, or not having more than only one sperm cell depositing the nuclear material inside the oocyte is for obvious reasons. Okay, you cannot have, or what you would have is like polyploid embryos, like 3N or so on. And that's obviously, in all of those cases, for the most part, this is incompatible with life. Okay, so probably those embryos would die, you know, right after fertilization. So you need to, at all costs, just allow one sperm cell to deposit the genetic material inside the oocyte. So the block is performed at the zona pellucida. Okay, so who triggers is, of course, by the time the sperm cell fuses, the membrane of the sperm cell fuses with the cytoplasm of the oocyte. When they fuse, that triggers a reaction in which calcium then go inside, or you have an influx of calcium in the cytoplasm of the oocyte, and that triggers then those cortical granules, you know, to basically fuse outwards towards the plasma membrane of the oocyte, and that triggers then a chain reaction by which you widen space, you harden the zona pellucida, you make the zona pellucida lose some glycoproteins, and all of a sudden, no more sperm cells are then able, you know, to actually enter the cell. So I think the last time we had somewhat of a picture here. So you do have the zona pellucida here, and you have the cytoplasm of the oocyte here. You would have like some of these cortical granules going all the way. You have very, very heavy sperm cell going towards, you know, the oocyte. At this point, this oocyte has to be gone through what process? What is the process that has to, this one has to be ready, it has gone through? Yes. So once you go through this capacitation process, this is ready, meaning
It's ready to tie with some of the receptors and glycoproteins that it's here in the zona pellucida. These two guys, or these guys, like these guys, they meet, they ties. All of a sudden, when this happens, what is the next stuff that happens then? After the capacitated sperm cell is bound then to the zona pellucida. Acrosomal reaction. So at that point, you're going to have, remember here, this little guy here has a little helmet. Okay? This little helmet is full of enzymes. It will digest this. It will propel the sperm cell to the cytoplasm, and all of a sudden you're going to have your nuclear material being deposited that will then meet the actual pronuclei or the nuclear material of the female. Okay? So when this happens, these granules here go, these cortical granules here go to the outwards of the oocyte, and all of this content will go then and is deposited in this space. Okay? You will see lots. I don't take too much of, in terms of the name, but, you have the zona pellucida, you have the oocyte. You can call it, in some books you may hear the name vitelline membrane or perivitelline space. To me, it doesn't necessarily, this is one of those things, I know it's quite technical, but to me, as long as you understand, there's the zona pellucida, there's a certain space, there is the actual oocyte, okay, the cytoplasm of the oocyte. And this space here, although it is not very visible even through the microscope, it is a very, very important space. Okay, you need that space in order to avoid polysperm. Okay, so they are released into this perivitelline space or vitelline space, and there is a reorganization of the zona. There is kind of a hardening of the surface of this perivitelline space. Basically speaking, not only biochemically, but physically, there's no more ways for this sperm cell to actually enter, the oocyte. So no more genetic material will then be deposited. Again, lots of little things. All of those little tables are actually very, very nice. So go give a good read on those. It's basically what we've been speaking in class, okay? So all the way then, once that happens, theoretically, and this is one nice thing about, and now let's jump a little bit to the applied side of it. The zona pellucida, once you have, for example, this little breach here that the sperm cell went in, this is a very microscopic breach. I know everything there at that point is microscopic, but it's a very tiny breach. It's not that you don't have, because of that little hole there, the zona pellucida is all of a sudden useless. No, you're going to have this guy here, and let's bring this guy to a smaller kind of side. You're going to have these oocytes that is now, let's say you have the two pronuclei, they're getting together. Once they get together, chromatins, the intertwine of the DNAs, all of a sudden you have your first diploid cell. Once you have this diploid cell, then this little structure is called no more oocyte, but it's called a zygote. Okay. How different that is from the previous oocyte? Well, aside from the nucleus, which is now diploid, the rest is very similar. Obviously, there's going to be lots of little changes that will go on, but up to that point, it is this structure here. From an application point of view to technologies that we use, not only in humans, but obviously in much more even in animals, is that up until the stage where the zona pellucida is intact, you can manipulate this embryo or this early oocyte of this embryo quite a lot. Okay, so you can go there and you have a little pipette, and you can bring up and down,
and you can transfer from one little tube from another without damaging the actual embryo, because the zona pellucida protects it. Okay, so lots of the technologies that we use today, for example, embryo collection or in vitro production or in vivo production of embryos or transferring of embryos from one animal to the other, is all done up until a certain stage where this zona pellucida is still intact. Okay, it is that important. So at that point, that's where we go into like some of this duration or events or progression until the first embryo cleavage. So we went on all this portion here. There's a little graph here in terms of when things happen. We are here at the first cleavage. Obviously, a few hours after, we have the actual intertwine of the two pronuclei. You're going to have the first cleavage, okay? And the first cleavage is nothing less or nothing more than just the same space with basically no longer one cell, but two cells, okay? This is the first cleavage. From a research standpoint, what we try to do when we develop or when we try to evaluate certain treatments and how that do with, for embryo development and even fertilization, we're trying to always look at this first cleavage. Why? Because if you have done something that improves that kind of process, it means that lots of little things have had to go right, up to that stage. Up to this or after this, we're gonna go like 2 cell, 4 cell, and actually in this very first stages of embryo development, that's really how you call this embryo. It's a zygote, then it's a 2 cell, then it's a 4 cell embryo, then it's an 8 cell embryo. By the time you reach a 16 cell embryo, you start calling this thing a morula. Okay, morula is just the name because it's a combination of cells about the same size, and it forms a little ball. Okay, you go on, and it really, really looks like this. You have the zona pellucida here, still the same zona pellucida that was, over there, used, for before fertilization. That's why I said that, I did some research before, and you could see actually how many sperm cells have actually attached to the zona pellucida, because, up to basically 7, 8, 9 days later, the zona pellucida is still going to be there. Okay, so that's how long that happened. So after this kind of stage here, you have this, before, I think you asked me about the twins and what exactly happens. At that stage, if you come with a scalpel here and you do section and you create 2 little balls, they can indeed go on to form 2 identical twins, okay? Sometimes that occurs just by you trying to play God, or other people, or naturally that can happen, okay? So which basically means those cells are 100% undifferentiated at that point. Okay? They can be transformed, they can differentiate into basically everything.
Is this splitting because you split the zona pellucida?
Well, no, not really. At that point, you would still, because the zona pellucida, you'll see here that, for example, let's say you're going to reach this stage here by about 5 days, maybe 6 days after fertilization. You're going to reach that morula stage. The zona pellucida will actually hatch or break about 9 days. So you can actually reach that twinning before. The only thing is that they would be still under the same zona pellucida. I think that's the only thing. Maybe the zona pellucida may hatch a little earlier. Okay, all it does, the zona pellucida up to that point, is really to protect, the physical protection kind of thing.
Now, let's see, some time of events, and normally this is we have talked about, some of these longevity in terms of, how long the sperm cell lasts, how much the egg lasts. Pay more attention to the range. Sperm cell tends to last a lot longer than the egg. It can go, if you say, yeah, it's about a day or so here, it's about less than a day, for the egg. To me, it's enough. It just means that, they don't last long. The sperm cell has a little bit more leeway there. They can survive a little more, but that's it. I'm not going to say, oh, but Ronald, what happens to the horse? Yeah, it's different. Okay, so at that point, it doesn't really matter for us. The embryo development, you'll see that, 2 cell, 4, 8. We didn't even have the morula here, but the morula here is in between. It's about 5-6 days. Blastocyst, and this hatching. And this is really the technical term we use to when the zona pellucida breaks. Okay? Because it's kind of an egg. So it hatches, and then you'll see. And hatches and does what? What do you think? So over there, up to that point, it's really a combination of cells. Early stages, you have this combination of cells. And basically, what you have to pay attention here is this word here that we call elongation. Okay, and say elongation. So it's going to elongate, and basically that's what it is. So it goes from this circular shaped form to something elongated. Okay? We all at some point in our lives will look like a little tapeworm, okay, inside of our moms. Okay? And why is that? Normally what happens then, what is elongating is not the embryo proper, not the embryo itself, is the placenta being formed. Okay? Because the placenta has to cover some surface area to then attach to the uterus. Okay, we're going to go through that, but that's the whole point. One interesting thing here that I said, from an application point of view, having the zona pellucida is important because you can manipulate stuff. The other thing that is important is this blastocyst transport to the uterus. Blastocyst is basically, I'm going to explain later, but it's the next stage after morula. Okay, so it's just the next following stage. How long does it take to reach the actual uterus? Okay, and you're going to see that, it may vary here in terms of hours, but overall speaking, you're going to see something like 4 to 5 days overall. Why would this be important for application? One interesting thing is, yes, up to 4 to 5 days, all the development of the embryo occurs inside the oviduct. Okay, it's not even in the uterus. So by the time it reaches that kind of a morula stage, it's already leaving the oviduct and arriving in the uterus. Okay. The second part that is important, and this is again for the application the same way as zona pellucida is important for us to manipulate, is that if you're trying to do something with an embryo that is kind of, produced inside of you, let's say you want to take that embryo to transfer to somebody else or to another animal, you have to wait 4 or 5 days or 6 days after the fertilization in order to collect that. It's very difficult for you to collect stuff from the oviduct. If you guys saw in lab, the oviduct is that very, very tiny kind of duct. If you want to kind of flush and collect that embryo from the uterus, you have to wait. And the reason is because it's only then that new embryo will actually reach the uterus. Okay? Make sense? Then we're going to go on to say when placentation starts, and then obviously gestation length. All of those dates and times and so on are important. Yes, they vary among species, of course,
But the most important part is that you understand that you are truly going from this to something that looks like a little group of little balls. The next stage, then, that's this portion, from there to placentation and so on. It is the portion that I told you before that we will touch base very lightly, but it's embryology. Okay, then there's pure embryology. You're gonna have to go from this to a full organized being. That's why lots of people do not understand, or we cannot artificially create another being. It requires an unsurmountable amount of cell communication and things that we have some idea, but we don't understand fully. Okay? This making sure that one cell then becomes a neuron or another cell becomes the tip of your finger, it's a very complicated thing. Okay, so people still don't understand. What we do understand so far is like, yes, eventually this thing here that we call the morula. Okay. That thing that we call the morula will then become something else. So a 1 cell, a 2 cell, 4, 8, 16. Here you can call maybe an early morula, a compact or not type morula, but a mature morula. And then they go into, and this is actually pretty similar in all species. They go into this pack of cells. Eventually this pack of cells, they kind of become like, very tight, very concentrated into a hard ball. But eventually this hard ball starts to expand a little, and then eventually a little cyst, a little hole, it starts to open inside of that embryo, okay? And this hole tends to grow, 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 and all of a sudden you start creating like cells that are starts to really go around the zona pellucida here. But within that expansion, you end up having, maybe in your slides you're gonna have a certain number of cells that are like, one side of this hole is a little thicker. What basically happens then is the following. These cells, and this is the point during the blastocyst stage where you have the first set of differentiation of cells. So you do have again, the zona here, and you say, okay, the morula was this tight amount of balls, and then you have the blastocell or this whole thing growing up in the middle, and then you have like lots of little cells here, in the periphery of this embryo. And you have a few other cells here that form around one of the poles of the embryo. You have the blastocell. This is what we call a blastocyst, okay? A group of cells, compact, we call morula. The next stage on with the hole in the middle, we call blastocyst. And this is the stage at which, for example, it would be more difficult for you to do the splitting, okay? Because then structurally they are already starting to have a certain shape, certain form, okay? Does that make sense? In this, that they have enough to. What are these differentiation or not? So at this point you call this cell here or this group of cells inner cell mass. Not very fancy or anything. It's just an inner cell mass. That inner cell mass is what's going to be the embryo itself. Okay? other cells here in the periphery, we call them trophectoderm, okay, or trophoblasts or trophectoderm, whatever you want to call, like, but it is, you can hear different names depending on where you read. But these are the stuff, this is the stuff that will give rise to the extraembryonic membranes, okay? And you have heard this word extraembryonic membranes somewhere, either studying, in embryology, people love to study birds, you know, to study embryology because, you know, it's inside of an egg and you can see all the whole thing happening, da, da, da. And if you remember and you have seen those classes, you'll see that do these names ring a bell to you? Allantoid, Corium, you know, Chorioallantoid, and the, you know, there's all this what we call extraembryonic membranes because they are membranes that are not necessarily part of the embryo, but they grow around the embryo for various reasons. So if you see embryology in birds, you will see that some are, or the some of those membranes or some of those sacs, they serve for nutrition. The other ones for excretion. Some of them for both. Some of them more for, you know, physical protection. Okay. Anyway, so there you go. So this would be the time where you have that little picture there. Hatching. By about 10 days of development, you have the actual hatching of the blastocyst. So by then, you basically no longer have this physical barrier that holds the thing into a circular shape. Okay, so these no longer have to stay in that shape. Ronaldo, why 10 days? Why not, you know, somehow each species will have a certain day because up to that point, what you can really see is that, and imagine the green board is the actual lumen of the uterus. Okay? So basically what you're going to have to do it is, and in many species, you're going to have that kind of like cornuated shape. So it's kind of a tube. Basically what's going to happen from now on is that this embryo will elongate. We'll have to go from this format to something that is much more spread, okay? And it will actually spread through the whole length. How? And they do it quite fast. Yes.
Can you like detect that a species is pregnant by this time?
No, but that's a good question, and we're gonna get there. In no time. But it's excellent question. Perfect. So this stuff is, imagine this is growing. And this is becoming like a little tapeworm. And we've done studies where we collect embryos at day 15, 16, 19, and so on. And it is amazing. You're going to have this thing growing from maybe 2 centimeters in cows by the time it's like 14, 15 days to about half a meter by the time it's day 19. And they grow that fast, but they really look like a tapeworm. They really do. It's like a little long thing. So, Ronaldo, is that the whole thing, the embryo? No, all of that is basically the extraembryonic membranes developing. Where is the embryo at that point? The embryo is in that class of embryology, and they are going through ectoderm, mesoderm, endoderm, and formation of the notochord, the somites, They are becoming, do you know those pictures of, you know, little embryo that looks like a little fish, like have a little head and like a little tail, and little T-Rex lambs, you know, that, you know, they're really tiny. That's where the thing is at that point. And when I said that, not at that point, but once he elongates, this is what's starting to happen. Okay. Imagine that, you know, all of this, and let's say I said that that thing occurs at day 10. I said that the little half a meter embryo, it's on day 20 or day 19, something like that. up to that point, and then go back to the class where I was explaining endometrial glands. You know, how important the endometrial glands were because, you know, they have to produce, you know, some of those molecules to sustain the growth of this. Again, up to this point, there's no attachment. There's no placentation. Okay? There's no physical thing binding this extra embryonic membranes that will form through this ectoderm into the uterus. So the next question you should ask is like, so how the heck this little tapeworm survives? It survives through the product of these endometrial glands, you know, that comes out of the uterus. They produce stuff that goes into the lumen, and it feeds off, you know, this little tapeworm. I'm keeping saying tapeworm because they really are, you know, but it is that much of importance to it. Oxygenation, food, you know, nutrients, gas, everything has to be there, okay? So then you have, you know, for this class, you have a series of slides that we're going to go through swine, you have some of the cattle, some of the primates. It's not that you have to understand exactly what each of them are in terms of size and so on, but it will always go something like this. So yes, this is duration of estrus, that's fine. Gestation, all those two things is more curiosity. Go back to your other classes and, you know, try to match this with what we have learned. And blastocyst, hatching, ovoid form, filamentous form. And when I say ovoid form, yeah, it goes from a circular to kind of a ovoid to kind of a tapeworm. Okay, and this obviously put this in perspective in terms of size, but it goes on slowly, you know, growing and growing. Okay, yeah.
Are we expected to memorize the specific numbers,
No.
day-wise and size?
No. I think here, the basic thing that I want you to guys, but it's a good question, that I want you to get from the slides is that how fast it grows. It goes from basically 0.5 millimeter to about 1,000 millimeters in just what? 10 days. I know they're small, but it's fast, okay? And this is, I think this is the kind of stuff in terms of application that I think is important for you, and I hope for this class you take it, is that although it is small, the metabolic process or the metabolic rate of this little tissue is super fast, but you also can understand, I think up to this point, one of the things that I want you guys to take it not only from the side of the estrus cycle with all that CL formation and ovulation and synchrony for fertilization, and then early embryo and hatching and growing so fast. Do you understand how many things can go wrong during this process? That's why reproduction is so inefficient in nature. You have lots of embryonic losses in nature, okay? Why? Because it depends on so many little things to go right, you know, in order to have a fully grown, you know, embryo and fetus, that for the most part, you lose a lot of them, okay? So it's important to see all these numbers. No, it's important to understand how fast it goes. It's important to understand that, you know, yes, it goes from 2, 4, 8, morula, blastocyst. The good thing about the blastocyst is first time it differentiates into something. After that, you have hatching, and then you can say it's a, you don't need to even necessarily say, oh, it's a filamentous form or ovoid. What I want you to understand, it has to elongate. Elongate is a key word then for you to memorize. We're gonna go through this part here. To all the other, I think that the last portion of this class is actually maternal recognition of pregnancy, because in a little bit, we're gonna be going and basically meeting embryonic development class with luteolysis in a few minutes. Why this, how this, for goodness sake, who knows what the hell is going on? And then you go, what's going on is that, do you remember that time when I was saying to you that if the animal don't get pregnant, you return to the cycle, and you go to that GnRH, FSH, follicle growth, da 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 da. But now, what if the animal or the female is actually pregnant? If it is actually pregnant, something will have to happen for that cycle not to happen again. Saying progesterone goes down, da da da, because the whole thing about the embryo and gestation is that CL, that corpus luteum, instead of regress and another cycle eventually starts, that corpus luteum will have to last another 6, 7, 8, 9 months, depending on the species. The very same guy. So how does it happen? It will happen eventually. Going back to when the embryo is in this form here, this filamentous form, okay? Lots of this will happen before even you have a placenta and something else. And that's the freaking beauty of reproduction, okay? You have some of those like super clever mechanisms by which, the female find a way and the embryo find a way to communicate between the two and find a way for the cycle to continue and to have the little alien grow in there, and the little alien is not going to be rejected by the mother. Again, another layer of complications on how much loss does it occur, during the gestation and the creation and development of this offspring. So let's go back here. This portion here, yes, for the swine, estrogen production causes uterine secretions. Okay, nice. Ronaldo, do I need to know this part specifically and all those things? Yes, for one reason. As we move on to the other slides, and you're going to see one later, every species, and I'm not quite sure how every species got to the way, of indicating how for the baby to indicate to the mother they are there. Almost every species have a different strategy. Do you remember what is the hormone that causes luteolysis? Prostaglandin. Okay? So prostaglandin causes, and this is really the molecule that probably causes luteolysis in most species. But the way by which every species block the production of this luteolysis or the production of prostaglandin that will then cause luteolysis is actually very different among species. In swine, they produce a certain kind of estrogen, or estrogen sulfate, this is the name. And that causes the production of prostaglandin to, instead of going, for example, to the circulation, it goes to the lumen of the uterus. And that causes the CL to survive, the corpus luteum to survive. Pretty clever, but if you compare it to humans, to cattle, have nothing to do. Like all of them are different. So this is one. Allantois, fusion of chorion, allantois or placenta, this is basically saying, yeah, by day 18 to day 70 to day 30, or basically by day 30 or 40, the placenta really starts forming, okay? What is chorion? What is allantois? These are the extraembryonic membranes. For you guys, if you tell me extraembryonic membranes, that's enough, okay? But I just want you to recover some of that information from your previous 3 years of UBC, that eventually you can record that, this is what we're talking about, okay? Okay, so let's go back. That's okay. Let's go to the cattle. Filamentous form, blastocyst. Again,
It goes all the way from a certain millimeter to all the way here that has, in this case here, it has about conceptus occupy the entire gravitic horn. So it basically has anywhere about, and when I say occupies, it occupies like this. So it's much even longer. Again, the sense of growth, it goes from about 1 or 2 millimeters to 4 to 5 to half a meter. It is just super, super growth. Does it increases a lot in terms of mass? Yes, proportionally, yes. But by that time, yes, it is really, really tiny. It is still really small, okay? But it gives you that sense. One thing here that, in swine, obviously because you have several offsprings, I think you do have somewhat of a sense that once the sow gets pregnant with several fetuses, it's going to occupy almost everything inside of the uterus, okay? And here in the bovine slide, I said that, yes, by the time it is 30 days or so, you're going to have almost one entire horn being occupied by the placenta and so on. And this word occupation of space, it is actually rather super important. Why? Because now go back again to this mechanism by which the embryo will indicate to the mother it is there. Physical occupation is key for that maternal recognition of pregnancy, to say. Why? Because the communication at that point is not a classic endocrine communication, which means, is there a way for the embryo to send something to the bloodstream of the mother that then will indicate? At that point, there is no such a thing. There's no umbilical cord at this point. There's nothing, making that connection. So everything, and when this thing here grows and it starts to communicate with endometrium, the communication will have to be what we call paracrine. It has to be in a paracrine fashion, which is cell to cell, okay? Not cell, bloodstream, cell. It's just cell to cell, okay? That's okay. So this portion here, the conceptus produces quite a few things. Do not worry about this. The embryo produces something else there, or produces some testosterone or prostaglandin. It will all be important for placentation, but for us, the most important part is that what the embryo produces to block the secretion of prostaglandin later. So for the swine, yes, lots of things that are produced by the embryo. Estrone, or a type of estrogen, is the important part. In the case of bovine, and I don't know if I have this in the next, I apologize, I think I made a mistake. Maybe didn't include, but it is actually a cytokine. It's not it produces a cytokine by the name of interferon tau. This is the name of the molecule that the bovine conceptus produces at that specific time point that blocks, we're going to have another slide with that. But just to let you know, it's a very different molecule, okay? And I think, one of the things here for the primate, and we're gonna rush through the primate, slides. But one thing that, you do have in those slides is the following. What is different between primates and humans compared to the other domestic species that we're talking about is that they have the process here of menstruation. Something that the other species do not have. And menstruation is basically the shedding of this superficial layer of the endometrium, okay? And by the time you shed this thing off, or shed the endometrium off, you do have quite a bit of bleeding. And that's where, most of the bleeding occurs during menstruation. It is caused by a bunch of things, but the most important thing is that your estrogen or your, basically this proliferation, what causes this proliferation of the endometrium is estrogen and progesterone, but eventually what you have is a pretty sudden drop of some of these hormones, and all of a sudden you do have basically necrosis of that tissue. And by having the necrosis of that tissue, eventually is going to escape to the external environment. So let's go back here to the, where is this here on the luteolysis? No, implantation. So the other ones, basically what you do have for some of the other slides is a few examples. So I know this portion of the slides can be a little confusing, but the only thing that I want to take all the way from the swine slide all the way to the primate and some of the one from the dog is, yes, you have the growth. The growth is fast. It goes from a circular shape with the zona pellucida, with a filamentous shape, that tends to occupy most of the gravidic horn or the uterus, that side. It is important at that point, during the filamentous stage, that we'll be producing a certain molecule that will then block the production of prostaglandin, or at least that's the idea. That the molecule that is produced, it is different among species. So we gave two examples. One is the swine, which is estrogen sulfate. In the case of bovine, is interferon tau. In case of humans, is human chorionic gonadotrophin, HCG. Probably you have heard at some point about this hormone. So all of them, they have different strategies. And the overall strategy is to block production of prostaglandin. Only after that is that you're going to then do all of these extra embryonic membranes, which we mentioned, chorion or allantois. They will really start to develop and grow. And then placentation, this is what we're going to start talking soon. Placentation then will occur. Before we give it a quick break, just answering your question about when you can actually detect pregnancy. Remember when you say, you have this this filamentous kind of embryo, and let's say you have here the whole wall of the uterus. Okay. What I was saying is that the embryo itself is developing somewhere in here. The rest of the stuff is becoming the chorion, the allantois. They are growing at some point. At some point, as I said, you're going to start produce something that will indicate to the uterus to block the secretion of prostaglandin. Okay, so that's what I was talking so far. On your case specifically, and when you can actually detect cows, I always talk about cows, but animals that are pregnant, it will have to do with the amount of fluid that you start accumulating inside of this membrane. So if you see a picture of a fetus, you're always going to have the fetus enveloped in lots of membranes with lots of fluid, okay, and you have more and more. Eventually, that amount of fluid, at least for domestic animals, especially the large domestic animals, you can do this by palpating. You go there, you do palpation of the uterus, and all of a sudden you can feel that the uterus is filled with fluid. Okay? And eventually
You can even detect where exactly this thing will start growing more and more, and you can detect where this vesicle is. If you cannot palpate, as obviously, you know, if you have species that are too small, like such as the dog, or even humans, what do? You do ultrasound. So ultrasound then it would be a very good way to detect or to diagnose pregnancy, simply because it is very good at detecting what is fluid and what is not. You know, so as you pass the ultrasound through the belly, you're going to be able to see very well that, you know, the regular uterus normally would have no fluid inside. By the time you become pregnant, then you have that uterus filled with quite a bit of fluid, and you do have very, very clear membranes around. And eventually, at some point, it grows well enough where you start seeing the embryo itself, bones, and so on.
How does like human pregnancy tests work? If we're doing ultrasound.
So the human, yeah, no, very good. So the human pregnancy test is a different thing, because we do have, for example, early pregnancy tests for other animals too. Basically, is that this guy here will actually produce a few things. In the case of the bovine embryo, for example, I said it produces interferon tau, but it produces also, once placentation, or this thing here, meets this thing here, when this embryonic membrane meets the endometrium, this is when you start talking about placentation. Placentation is nothing less than the actual physical attachment, you know, from the mom to the baby. In the case, for example, of bovine, once you have that initial attachment, let's say 20-something days, you're going to have a differentiation of certain cells here, and these cells produces a few changes in or a few different molecules. So in bovine, we have one that we call a pregnancy-associated glycoprotein. Okay? And there are several types of this protein, and this goes into the circulation. So people have found out a way to create antibodies against this PAG protein, and all of a sudden, you can probably today detect by 24, 25 days. It's not too much before ultrasound. Ultrasound by 25, 26 in bovine, we can actually also detect, but much earlier than manual palpation. Okay, so this would be one way. In the case of humans, it's probably even more like you can have, what's the earliest test you can get today?
2 weeks.
Which is just ridiculous, like it's crazy how early that is. There's two things there. By 2 weeks, so this would be more or less, you could say, the time when, you know, some of that maternal recognition might go on as well. But over there, I think, I can be mistaken, I think it is exactly the stuff that is produced. But one thing that I don't recall if it is HCG or some downstream protein from HCG. I don't recall now. I'll look that for you. But it's something with that. So it'll be a protein that can actually detect that specific protein. Because this, in humans, if I'm not mistaken, is actually this actual protein is that what's going to go to the corpus luteum and actually protect the corpus luteum against prostaglandin. Okay, so I think that's the one. Another way in which you could do as well is, do you remember the next portion of this class, at some point we're going to talk about immunology and reproduction. And we talk about a little bit about immunology and reproduction because I mentioned before, you do have the little alien growing inside of you. So what does the body do about that? how don't we get rejected during gestation? And in some species, so for example, the bovine, I said that interferon tau is the one that is produced here, but then eventually, but interferon tau, for example, is a cytokine. It's a molecule that is produced when you have viral infection. Okay. Why did the nature chose antiviral molecule to be the signal for maternal recognition of pregnancy? I have zero idea. I do not know. But what I do know is that in many species, you do have certain changes in some immune cells inside of you during pregnancy. And for example, one of the ways today that people have found, even for the bovine and people, some of my colleagues are studying how you detect pregnancy, is actually by isolating some defense cells, and you do gene expression of these cells, and all of a sudden you see that these cells are different in a very unique way, and that indicates pregnancy. Who would know? Okay, so who would know that that would be the case? But that's really how it's done. But in general, it is either the molecule itself or a downstream from that molecule. And the only thing is, talking about then, that gestation, that different gestation diagnosis. So physically, you have to have fluid from either from a ultrasound or a palpation standpoint. From a molecule point of view, it has to be when the embryo produces something. And let's be clear here before we move on to placentation, and we're going to go into other parts. Up to this stage, this big ass tapeworm, okay? Up to that point, the mother does not know there is an embryo there. You see, he ovulated, he has all this stuff, and during the estrous cycle, do you remember, up to that point of luteolysis, up to that point, the length of the estrous cycle of any species that goes from the beginning of the cycle, or in that case, it would be the fertilization, more or less, up to that point of luteolysis, there's no way or no difference for the mother. The mother is functioning, or the uterus even, is functioning at exactly the same way you would function in any other estrous cycle. Everything is the same. That is the only point, or the first point, where you're going to have, okay, something is different, and there is something that is biochemically will be different in the mother. That's why we call that moment maternal recognition of pregnancy. Because that's only when the mother recognizes that, there's something different inside of the uterus. And it's probably the earliest you can ever be or ever get to diagnose pregnancy. Before that, you would have to do, they're too small to physically detect, even with ultrasound, even with MRI, too small, it's too tiny, don't have anything going on. Now, the earliest would be then to have some sort of protein, some sort of molecule that you can actually detect. Does that make sense? Yeah. Okay, I didn't shut up, so let's continue. So this is okay. So this is the we're gonna go back to maternal recognition of pregnancy, but I think it was important for that to mention that. Now let's go to a little bit of the physical part. So we're saying that this thing is growing so fast, it is occupying the horn, but then next step, and we mentioned here a few times, placentation. Here we go, placentation. When you, for example, and this is a question, maybe you guys don't have this question, but this is a question I used to have when I was in school. If you have to picture a lung, can you picture like the lungs? You say, okay, it looks like the heart. You say, okay, the heart looks like this. The intestines, It looks like this. The kidneys it has that bean shape, kind of. What about the placenta? Can you describe to me how the placenta looks like? We call the placenta. Oh, we say the placenta. Oh, the placenta. Yeah, it goes. Yeah, right. What does it look like to you? Oh, I saw in a movie, you know, after the birth, the placenta comes out. It looks like something, a blob. What do you think? Does that make sense? You know, we don't have much of this idea of what the placenta is. And in different domestic species, placentas will have different shapes and forms. The placenta ultimately is what? Is basically the connection between the mother and the embryo. Okay, it's the physical connection between the two. Because we have very different types of placentation, the placentas will look differently. And the placentas, the only thing that is true is that the placenta will always be tied with the outermost embryonic membrane. Okay? Why? Because the outermost is really the one that will attach to the endometrium. Does that make sense? No? Yes? Sort of? Yeah. More or less? Okay. So if you have to have this, and when I say the extra-embryonic, so this is one of those embryonic, extra-embryonic membranes. So it's growing here, and let's say it's growing more, then it's growing more, and it's growing up to a point where it physically connects to this. Okay, so this is the endometrium. And this is one of the extra embryonic membranes. Okay. So the connection between the two is actually what's going to give rise to the placenta. Okay. You have to understand that, you know, before the action, and when you say, you know, what do you mean by connection of the placenta? Is the placenta the umbilical cord? Is the placenta, do you know what that picture is? You see the umbilical cord, and it's beautiful. You know, it looks like a little, you know, it's in a little blob, and it's full of vessels and going around. That thing is a portion of it, but it's not the placenta itself. What it is just the placenta getting all the vessels from the entire placenta to one point, which is the umbilical cord. Okay? So it's just the connection, okay? The physical connection of the vascular, you know, irrigation of the placenta. But the placenta is almost something like this. For most of the species, what you're going to have is something like this. This connection can occur in different ways. Okay? So in cows, you have connections like this. You have one here, and you have a space, and then you have one here, and then you have space, and you have one here, and you have a space. And imagine this going all the way around. What would be the umbilical cord? Is the connection eventually will give rise to a little vessel. And there's a little vessel, there's a little vessel, there's a little vessel, and eventually they centralize into something that will tie to the embryo. The little section that ties all the other vessels to the embryo is the umbilical cord. Okay? But in many ways, all of them will work like that. The only thing is that you look at the placenta of a cow, it's like this. I'm going to show pictures later. If you look at the placenta of humans, humans tend to have all that connection in a little cup, and you call it a little disc. Okay? Why is it a disc or not? I don't know. It came out like that. Okay? If you look at dogs, dogs have, imagine this is the
the blob in which each dog is. And you have like imagine you have like a layer around, just like a little tape, around. And that's how the placenta basically organized itself. So you have this, and that it is true. When I ask you the question, do you know how it looks like? You wouldn't know. Every species will have a different format. But the basic premise here, and it's okay, I'm not going to ask you what types of placenta, in terms of basic or macro morphology looks like, but they have different ways of organizing itself. What it is important here is that you're going to create that physical connection. And that physical connection will tend to be quite, or rather, disruptive to the endometrium and to the cells of the embryo. Why? Imagine you have, as I said, the tapeworm, the endometrium. They're going to have to, first of all, have the physical connection or the physical touch between the two tissues. But eventually, certain areas of this tissue will have to transform in a way where they're going to go from this to something like this. And from this newly formed connection, you're going to have to form vessels, angiogenesis, and all vessels being formed. It is an extremely complicated molecular or cellular thing going on at that stage. But that's what you're going to have. So you're going to have cells invading each other's tissue and going and doing that. Now, before we go on, because we're going to be done with the class in 10 minutes, but do you understand why the immunology in reproduction will be important? Can you imagine a tissue that is not yours all of a sudden invading your own and your body saying, that's okay. You can do it. You're going to have a special type of relationship there in order for what? Rejection not to occur. Okay? You're going to have something different going on there in order for rejection not to occur. So implantation. So let's start with this word here. The implantation is this contact. You're going to have nutrient exchange. Yes, the placenta, not only doing this exchange, the placenta in most species will also produce some type of hormone. Okay? You're going to have invasion and interaction. The maternal recognition of pregnancy, this occurred for most species before. This thing normally occurs before the placentation. But then, obviously, the placentation itself is a little bit after this implantation. The word here, apposition and implantation, sometimes they use these words almost interchangeably. Apposition is basically the first step, is the actual touch, between the two tissues. Implantation is then the beginning of that little blurb that I said that, one tissue invading, the other. And from this apposition, implantation, then you form the placenta. Okay? And the placenta, where these connections occur depend on the species. In cows is like that, in humans is like that, in dogs is like that, and go on and on. Okay? But all of a sudden, what you do have is that, yes, most of the species, at the end of the day, they do have, and I normally cite the umbilical cord, because the umbilical cord is this visual thing that everybody understands. They see the umbilical cord. They all have umbilical cords, independently of the type of the placenta. Why? The umbilical cord is just the bringing all of those vessels that whatever type of placenta you have to a central location that then ties with the offspring, with the fetus. Okay? Does that make some, we're going to go back to this on and on, but does that make some sense? Okay? Isn't that beautiful? Isn't that amazing? No, seriously, it is.
to think about this, like how nature works to have this thing going on, it's just crazy. It's crazy that we're alive. We're all winners. Huh? Yes, we are. So this is, it starts like this. So one of the things that, I put it in this, and this is just a picture of this little thing, goes back to the, I think the analogy that we can make here in terms of touch or physical contact is the same as when we had like the sperm cell and the zona pellucida. The sperm cell and the zona pellucida, they can meet each other, but they will only attach if there are receptors that bind to each other. That depends on what? Capacitation and zona pellucida, maturation, and they go and they bind. For these to happen is also somewhere the same. It's not like anything, just because it is close to each other, we will attach. It won't. You will need something there. Part of the stuff like this PR here is progesterone receptor. Okay? This progesterone receptor is something like this. When you do have your estrous cycle, remember, you ovulate, and then you have the beginning of another estrous cycle, and your progesterone at that point when you're beginning there is low, but then it goes up, and it continues. Theoretically, for the gestation, this progesterone will continue a high forever, up until the end of the gestation, okay? The only thing that happens, and you're gonna tell me this, what happens when you get super exposed to the same hormone for long periods of time? What is one of the things that happen to the receptor if you're super exposed to the hormone all the time? And this is something that is general for almost all hormones. You downregulate yourself because of that. The reason why before you had, that pulsatile pattern that I said that it happened with GnRH, LH, and stuff, that normally happens to many of the hormones because the binding with the receptor tends to do what? Tends to bring the receptor inside the cytoplasm, and either it's going to be recycled or something else. But normally what happens is that you have a pulse, it binds to the receptor, your receptor levels in cells go down. But then you produce less, and then your receptor goes up, and then you have another bump in hormone, and you go like that. For lots of the hormones. The problem with some of the steroid hormones is that you produce that thing and there's no pulsatile pattern of release of steroid hormones. Estradiol, testosterone, progesterone. Once the corpus luteum is there, it keeps pumping progesterone. Okay? It keeps pumping progesterone. So what happens eventually in the endometrium is that you have progesterone receptors, and then you have progesterone receptors, but eventually, when you have this super exposition of progesterone, your progesterone receptors go down. As your progesterone receptors go down, it doesn't really matter if you have too much progesterone or not. The effect on these tissues somewhat, or at least temporarily, gone. And this is what happens. You do have some of these mucins here, this mucin 1, and this little glycoprotein, what it does is that it tends to, once you have the progesterone there, it doesn't allow the embryo, the elongated embryo, to bind to the endometrium. Once you have this progesterone receptor going down, these mucins, they disappear. Some of these glycoproteins, they disappear. And once that happens, the embryo then is able to bind some of these, what we call integrins. Integrins are very, very, very common attachment molecules. They are present, for example, in white blood cells. When our white blood cells have to bind to your endothelium to go from the bloodstream to the tissue during inflammation, for example, they have to have a way to bind
to the endothelium and going inside your body or the interstitial space. And this is what happens. So you allow some of these integrin receptors to surface in the endometrium. The embryo have some of these integrins. So all of a sudden, you have the first way of, or the first method that the embryo found to actually not only touch the endometrium, but to actually bind mechanically and also chemically, to the endometrium. Does that make sense? It's kind of a learn, but you're gonna get there. Okay? Okay, so let's just stop here because the next one is, I know we went on a little bit to the placenta. We're gonna go back to the placentation, but it is important that this portion of the apposition and then maternal recognition will cover more on the next class. So just one thing. For next, it's going to be, so next week is going to be Tuesday, no class, and then on Thursday will be, so next Tuesday, and I'll be gone for 2 weeks. So the only thing that will happen is the following. The week after, so next week, we're supposed to have a quiz on Thursday, right? And what we're going to have also do is like a review session with the TAs and also any help you'd like to have for your presentations on Thursday. So by the time you do the quiz, you come here, get the quiz, and you can have time with the TAs just to solve those things. Okay, so if you need tips on how to do the presentation, if you need more, although I did not understand anything that we have looked so far, the TAs will be here. Tuesday, we were thinking, oh, let's do this, let's do that, and I said, let's have a beer. Okay? So what I said is like, you guys are free next Tuesday. Okay? The week after is going to be all labs. Okay, so we're going to have another set of labs, and I think the next ones will be what? Technologies and
artificial insemination, embryo transfer, and then cytologies and biopsies.
Yes. So biopsy, cytology, artificial insemination, and embryo transfer. It is awesome. It is nice. You guys should be there. So the next 2 weeks will be like, labs and quiz and some other review. Try to use the Thursday especially to answer questions about some of the presentations and so on. Is that all?
What is the quiz on? Quiz cut off.
Come on. Up until now. No, I'm kidding. So the last one finished on embryogenesis. Don't ask me. I don't remember everything.
We didn't do any of the aspects or stuff.
Nice that you remember that. Okay, so that will be included. Let's see the next one. Okay, so puberty and estrous cycle, fertilization, yeah. Establishment of pregnancy. Establishment is this one, right? Okay, puberty and estrous cycle and fertilization. Okay, very good.